Calcium is the most abundant mineral in the body. Inside cells, it is used as a secondary messenger to activate a chain of reactions such as intracellular signaling, vascular contraction and vasodilation, nerve transmission, and cell cycle regulation. The average absorption rate of consumed calcium in the body is 30%, but this can vary during a person's life throughout periods such as pregnancy and lactation, when more calcium is required for milk production. The calcium concentration in blood plasma is tightly regulated by hormones. In the unlikely event of a sudden decline in calcium levels, calcium storages in the kidneys, intestines, or bones are utilized immediately to prevent hypocalcemia and maintain homeostasis. Due to their positive charge, calcium ions require channel proteins to go through cellular membranes. Calcium channels regulate ionic flow using mechanisms such as voltage gating and ligand gating, among others. Additionally, some channels are highly selective to the ions that they allow through, while others are not so much. Some cellular functions, including signaling, require the concentration of specific ions around their membranes to be built against their osmotic gradients and move towards certain threshold values. This is accomplished by ionic flow regulation through channel proteins embedded in said membranes, allowing membranes to attain necessary voltages. Negative feedback loops are used to prevent voltage differences from becoming extreme by having high concentrations of specific ions lead to channel inactivation. A specific group of channel proteins that regulate ion flow in cells are called transient receptor potential channels, abbreviated as TRPs. They play an important role in relaying sensory information and are most often found in cellular plasma membranes. Mutations in TRP proteins may also play a role in cancers and have previously led to skeletal dysplasia and neurodegenerative and kidney disorders. There are 7 different subfamilies within the TRP family, and there is high diversity in their mechanisms of activation and roles. The vanilloid subfamily is specifically highly selective to calcium ions. TRPV6 is one of the proteins within the TRPV subfamily. Its channels are open at basal concentrations of calcium in cells. But when calcium levels exceed these basal concentrations, binding of a calcium-modulated protein closes the channels. The calcium-modulated protein is known as Calmodulin, which is abbreviated further as CAM. CAM is a highly conserved protein found in the cytoplasm of all eukaryotes. It is dumbbell-shaped with 2 globular ends linked by an alpha helix. At each end, or also known as lobes, has 2 binding sites for metal ions. Calcium binding in these sites is thermodynamically favorable compared to binding of other metallic ions due to steric interactions. When calcium is bound to the CAM lobe, the ligands are separated by favorable distances. However, when instead an ion such as magnesium is bound, the ligands are forced to come closer together, which induces steric hindrance. So, due to its size, the binding sites have a greater affinity for calcium. With calcium bound, CAM undergoes a conformational change that allows the TRPV6 complex to relay signals to other proteins, such as kinases and phosphatases for ion transport. Our group read the article titled, "A Novel Mechanism for Calmodulin-Dependent Inactivation of Transient Receptor Potential Vanilloid 6 by researchers at the University of Leicester in England." The hypothesis presented here is that if the structure and inactivation of TRPV6, considering its complexation with calmodulin, are elucidated, then a model for calcium-dependent negative feedback mechanism of TRPV6 can be derived. In the mechanism presented, calcium acts as a secondary messenger to inhibit its own flow into the cell through a negative feedback loop. At basal calcium concentrations, 2 calcium ions bind to 1 lobe of the CAM protein, which is also bound to the TRP channel. Under these conditions, the channel is open. However, when calcium concentrations increase, 2 additional calcium ions bind to the opposite lobe of the CAM protein. This causes CAM to bind to the other TRP protein on the right side as well, closing and thus inactivating the channel. This mechanism explains how calmodulin is utilized in activating the TRP calcium-specific channel. Mutations in the TRPV proteins lead to deviations from homeostasis, such as hypercalcemia and hypocalcemia. Thus, the proper function of this complex is essential for calcium regulation in the body.